right. Um, First, thanks, Amber, for giving me the opportunity. Uh, what I'd like to do today is actually talk about something very different. So, just sit back, listen, participate. We're probably going to take you a little bit deeper into the whole concept rather than working on the classical pathways of drug discovery and development, which I've been doing for decades in my career. I kind of fell into this whole area of the mitochondria. This is something, just as a disclosure, I'm part of Blue Oak Nutraceuticals. We're doing mitochondrial work, but also at Ion Therapeutics, where we're also interested in a variety of therapeutic areas. Normally, one shouldn't do this, but let me ask the question. How many of you have good familiarity with the mitochondria? Yeah? People have heard about it. People have listened about it. But let me start by just giving you a bit of a brief on the mitochondria itself. Now, of course, it is the central organelle, and it's fundamental to life and health itself. Just some ideas, just starting from the left side. The only way we recreate more mitochondria is through exercise. That's the only physiological stimulus we know how we can make more mitochondria. And then the destruction happens through the formation of these permeability transition pores caused by a variety of insults such as high calcium, glucose, other oxidative injuries, including monogenetic diseases, etc., that results in cellular stress that destroy the mitochondria. The mitochondria typically have a half-life of 3 to 4 weeks, so you got to keep making more of them. But what stands is on the right. So most of us, as an adult human, we have about 100,000 trillion mitochondria in our body. That's a whole lot of mitochondria. The main job is to take nutrients and turn it into energy. This is the functional basis of how we live. When you breathe, move, blink your eyes, whatever you do, your body is using the mitochondrial energy to make that function happen. And goes through in the entire life. And they make ATP. I'm not going to go into the details, but it's adenosine triphosphate, which fuels all cellular activities. And this also includes translation of proteins, post-translational modification of proteins, transport, transposition of proteins, etc. And a healthy person at rest produces their body weight of ATP every day. So if you're 160 pounds, you're making 160 pounds of ATP every day, nonstop. That's a whole lot. At maximum exercise, when you exercise, you can be making up to half a kilo to a kilo per minute of ATP in your body. And this is essential because without ATP, nothing happens. The entire system comes to a dead stop. ATP must be produced every second of every day. It cannot be stored. So your body makes it when it needs it. It does not circulate it. It stays within the cell. And every cell uses about 10 billion ATP per day. So just imagine the number of cells in your body, and each cell is using 10 billion per day, which relates to about 10 to the 25 ATPs per day. What I'm trying to tell you with all this is that fundamental to our own life and health is this inherent process that is happening every second without fail. And every time this process is interrupted, results in consequential or causal pathologies or outcomes or comorbidities, whatever you want to call it. And it is the only component in your cell that can make more of itself when we need. So if all of you decided to suddenly get up and start running, your body will kick in and start the process of making more mitochondria. It will fix itself because oxidative damage destroys the mitochondria, and it'll fix itself. It'll through fission and fusion processes. And when you have too much of it, it gets rid of itself. So a cell has a homeostatic balance where it maintains a certain level of mitochondria, which is optimum. When it goes down, it makes more. When it has more, it gets rid of itself. Just think about it. Your average body temperature is 98.4 degrees Fahrenheit, right? It goes up to 99, that's 0.6 degrees Fahrenheit up, you are febrile. At 1.6 degrees Fahrenheit, you have fever, right? So this mitochondria is maintaining that body temperature exquisitely day in, day out, and it is balancing that constantly. How did it come about? I'll take you back about a few billion years. This is the ancestral prokaryote cells, no membranes, which leads to infolding on plasma membranes and the formation of the nucleus. And then there was the endosymbiosis happening through the aerobic bacteria and cyanobacterium. And this symbiosis led to what we know as the ancestral eukaryote, which eventually evolved into the modern cells we have, with the cyanobacterium splitting out into chloroplast that plants use, and these becoming the mitochondria that we use. Every living creature on planet Earth uses the mitochondria. Every plant on planet Earth uses the chloroplast. And this is the central fundamental mechanism how we produce energy. And it is rooted in the laws of thermodynamics. It creates a membrane potential. When that membrane potential passes through a particular protein, ETC synthase, you make ATP. Every living creature on this planet uses it. Without this, no life. So we started to ask the question, what is the relevance of it? So, in ancient Rome, all roads led to Rome. In my work, all roads led to the mitochondria. Every organ essential to all biological functions. There is no, whether it's the skin, we don't mention all of this, but critical, non-critical, every organ has the mitochondria. There's only one cell in your body that does not have the mitochondria, and that's the red blood cell. Okay? And the red blood cell is the only cell that does not have a mitochondria. Every other cell does. Okay. So, what about the mitochondria? The mitochondria, as I said to you, is a bacteria. So everybody has been telling, or we have this notion that we have a single genome in our body. Actually, that's not quite true. We have two. The mitochondria has its own genome. It's a very small genome, but it's a bacteria genome. And their pathology is across, hence the mitochondrial pathology is across two genomes. One is the primary mitochondrial disease. That means when you have a mutation in your mitochondrial genome itself. Those are the primary mitochondrial diseases. And there's the secondary mitochondrial dysfunction, where another pathology affects the mitochondria, whether it be any monogenetic disease, diabetes, tumor, infection, they all affect the mitochondria. And these are some of the pathologies I have listed, but of course, if I listed everything, it would just not fit on the slide. So the consequence of mitochondrial depletion and dysfunction is all these, and I put ... because I could go on and on. And listed amongst these are also these primary mitochondrial myopathies, but also related to neurodegeneration, heart, brain, aging itself. At the age of 21 is when we have peak number of mitochondria in our body. That's when we stop growing. Past age 21, all of us are losing about 15% of our mitochondrial density every decade. And that's aging. The entire aging process is associated fundamentally to the loss of mitochondria. Hence, exercise is the only way. But also, our lifestyle affects it. The food we eat, how much we sleep, what exercise we do affects the level of mitochondria. So let's talk about SLC6A1. Why am I telling you all this? So we understand from the functioning of phenylbutyric acid, et cetera, is related to ER stress. And we accept, and Zach talked about a variety of mechanisms people have positioned. Let me throw another one in between, and I'll get to that. And perhaps phenylbutyric acid is playing a role as what I would like to call an anaplerotic agent, and I'll come to that a little bit later. But clearly, the idea here you have is the translocation of the GAT1 to the presynaptic neuron, and then when you have this mutation, the GAT1 gets stuck into the ER. You don't have the reuptake process. But also it affects the astrocytes. And why is that relevant? Because this... Did I say something? Cool, thank you. All right, I'm going to not move anymore. So part of this is because this GAT1 here also plays a role, because this is how GABA is taken over, converted back to glutamine, and goes back to the presynaptic to be recycled. So this reuptake through the astrocyte is also important. But this level in the synapse of GABA, when you do not have this reuptake, leads to the activation of the non-neuro, the peripheral neurons. And this is where a lot of toxicity of the side effects, the comorbidities, arise from, where you lead to enhanced tonic inhibition and decreases things like cyclic AMP. And this leads to a process through cyclic AMP, which we know regulates mitochondrial fission and fusion, motility, mitophagy, but also autophagy, apoptosis, and mitochondrial protein import. What I'm trying to tell you is that as we have this inhibition of the reuptake through the reduced expression of GAT1 at the presynaptic neuron, the higher levels of GABA in the synapse leads to the activation of the pathogenic pathways associated with SLC6A1. And part of the consequence of that perhaps leads to the mitochondrial issues, and that manifests itself in the phenotype that we see. Because ER stress is associated with all these parts, and they're all mitochondrial, right? Mitochondrial DNA, reduction of PGC1 alpha, which is a cofactor for mitochondrial biogenesis, NERV2, electron transport chains, go into sirtuins, the mitophagy levels, mitochondrial biogenesis goes down. Calcium influx goes up, leads to more mitochondrial toxicity. So what I'm trying to tell you is that as ER stress happens, there's a direct consequential effect on the mitochondria. And perhaps that is a part we can think about. So this is how the communication happens. So these MAMs, known as mitochondrial-associated membranes, and this calcium influx goes in, and this leads to mitochondrial depolarization, fragmentations, reduced oxygen consumption, increased mitochondrial permeability transition pore, which is the prelude to the death of the mitochondria. In every cell, it's the mitochondria that decides when the cell will die. As long as the mitochondria are alive, that cell will not die. And if the mitochondria with the permeability transition pore releases cytochrome C, which then leads to the death of the cell. Mitochondrial dysfunction and seizures, so what I'm, in summary, is impaired mitochondria then disrupt the proper energy production within neurons, leading to altered neuronal excitability, which is increased tonic inhibition, increased susceptibility to seizure and activity. And when mitochondria cannot produce enough ATP, neurons become more prone to firing abnormal, resulting in seizures. And here's the dysfunction leads to ATP reduction, increased ROS, increased calcium, and seizures. And this turns into a loop. Now you have an energy depletion, defective homeostasis, tissue excitability, seizures, oxidative stress, dysfunction. It goes around and around. And this, of course, does not mean that every patient will have the same response. That response is now graded to the phenotype and governed by the genotype of the patient. And that's where you see heterogeneity in response. And the degree of ER stress matters. In the initial stages, you lose molecular quality control. Then eventually it's organelle quality control, and finally cellular fate and apoptosis. So as this ER stress increases, you see a higher degree of effect on the system itself. And this dysfunction can connect the diverse medical symptoms associated with ASD. And if you look at it, that all these clinical abnormalities found in mitochondrial diseases are also found in ASD individuals. So there's a correlation between what people see in primary mitochondrial disease, where the mutation is in the mitochondria, and secondary mitochondrial disease, which is a stress on the mitochondria. The clinical phenotype has a significant overlap because the causality is changing, but the consequence stays the same. The underlying part that also we need to address is inflammation. Chronic, subchronic, or acute inflammation is persistent, and today we have no way of treating it because mitochondria control the inflammatory process. They are the first gatekeepers to understand stress in the cell and induce inflammation. That is one of the first protective and responsive elements that happens in your body. And to this day, there is no therapy out there available that addresses chronic inflammation, even subchronic inflammation. None, zero. We may talk about all, genetic therapies, but today we cannot deal with inflammation. But through the mitochondria, you can. It's the fundamental organelle. If you manage it and you maintain its homeostasis in a cell, you can manage inflammation. And it's something that we have to start thinking about. So what I'm telling you, key points about mitochondrial dysfunction is energy deficit, calcium dysregulation, oxidative stress, and this is the primary mitochondrial diseases in epilepsy. So several often manifest with epilepsy as prominent symptom. This is primary, such as MELAS, altered neurotransmitter releases because of disrupted energy production, impaired synaptic plasticity, and that has to do with cognitive development, mental development, because plasticity in the neuronal development is a significant role in memory learning and development, neurological development. And of course, finally, it leads to neuronal death, which can trigger permanent programmed cell death or apoptosis in neurons, which leads to neuronal loss and further impairing brain function. So this is a spiral that you get into from the fundamental processes of dysfunction. So to close the loop, the question is, how does phenylbutyrate reduce ER stress? Is it an anaplerotic for the mitochondria? And it's a posit. So what is an anaplerosis? Anaplerosis is creation of mitochondrial citric acid cycle intermediates. So for all of you who are familiar or not familiar, just a primer on citric acid cycle, also known as Krebs cycle or the TCA cycle, is the primary cycle within the mitochondria that takes in nutrients and turns it into energy. Right? So when you eat something, that's where it goes. And this has a number of carboxylic acids that go around, and those are known as anaplerotic agents, such as alpha-ketoglutarate, succinate fumarate, and this is the basis of the ketogenic diet. The benefits of the ketogenic diet are directly related to the Krebs cycle, that these are the nutrients that enter the Krebs cycle and provide the nutrient that the mitochondria need to provide the benefits you see on a ketogenic diet, right? Now, of course, excess leads to ketoacidosis. So there's a fine balance, and diabetics suffer from excess ketones in the body, but that's not healthy. So these intermediates are interconverted rapidly, and the creation of any intermediate adds to the pull. One of the key ones is the beta-hydroxybutyrate, is an anaplerotic agent, and that's the structure of beta-hydroxybutyrate. It has this 4-carbon ring chain with this hydroxy and carboxylic acid, and it feeds into the Krebs cycle, and it makes the intermediates through internal biochemical synthesis. Goes through the citrate cycle, acetate and glutarate. So is phenylbutyric acid also acting as an improved mitochondrial function and reduces ER stress as an anapyrotic? And what I give you is one of the metabolites of phenylbutyrate is this. And I'm sorry, I have an extra carbon here. As a chemist, I should know not to do that, but it's not 5 carbons, it's 4. So this carbon should not be there. So no, it is 1, 2, 3, 4. I'm right. Okay, so this is fine. I'm seeing things wrong. So it forms this beta-hydroxy acid, which has a very close similarity to this, except in this case, there's an extra phenyl group, right? So if this didn't exist, this would be the substrate. And the question is that, is it sufficient to provide benefits to the citrate add cycle? But it's a poor mimic. Perhaps that's why we need the large doses we're giving, that it is not a good mimic of the butyric acid. And last talk I heard Dr. Kavanaugh give was he was looking at butyric acid and found to be a much better analog to have the effect, right? So there's a structural correlation and study that we're seeing that if you take butyric acid, which makes the required intermediate, the keto acid, it functions better in removing ER stress than phenylbutyrate. And so the question is that, are you making an analog endogenously through metabolism that is more helpful? Good question. I'm sure we'll hear more about it from Dr. Kavanaugh or others. But it starts to make you think that perhaps this is a potential role that phenylbutyrate is playing as an anapyrotic agent to supplement the mitochondria. The issue comes in is they are the fuel, but what about the furnace? The issue is that, yes, you can give more anapyrotic agent with the fuel, but if you're fundamentally losing the mitochondria, do you have enough furnace left to burn the fuel? And so the question to us always is, how do we work in ways to supplement the mitochondria itself, not just the fuel that we give? So with that, I will end, and thank you much for listening.